Welcome, everyone. Today, we are diving deep into good laboratory practice, or GLP. This isn't just about regulatory checkboxes or passing an audit. It is about establishing a rock-solid foundation of data integrity that ultimately safeguards human health and ensures patient safety. Over this course, we will unpack how to transition standard laboratory workflows into audited excellence, exploring the core pillars and historical lessons that shape our testing environments. Before we dive into the technical regulations, we need to address a deceptively simple question. What actually is GLP? Think of GLP as the operating system running silently in the background of every compliant laboratory. It's an integrated framework engineered to guarantee data reliability. Now that we know GLP is a quality system, let's see what this system actually governs in practice. Next, notice the phrase organizational process and conditions. GLP doesn't only focus on laboratory experiments. It also considers the people performing the work, the equipment they're using, the facilities they're working in, and the documentation supporting every activity. The official mandate of GLP applies strictly to non-clinical studies. Before any drug candidate is administered to human volunteers in clinical trials, it must undergo rigorous non-clinical safety testing, such as toxicology and pharmacokinetic profiling. If we mess up non-clinical data due to poor lab habits, we risk making flawed decisions when real human lives are on the line. That's why strict GLP adherence at this non-clinical stage is absolutely non-negotiable. Many people assume GLP begins when laboratory testing starts. In reality, it begins much earlier, with careful planning. A well-designed study reduces errors before they ever occur. As the study progresses, every activity is monitored, documented, and eventually reported. But notice that GLP doesn't end when the final report is signed. The study must also be archived so that years later, regulators or auditors can reconstruct exactly what happened. Imagine trying to solve a mystery after all the evidence has disappeared. That's why proper records and archives are just as important as the experiment itself. Imagine two laboratories testing the same product, but arriving at completely different conclusions. Which result should regulators believe? Without standardized practices, it becomes almost impossible to determine which data can be trusted. GLP creates consistency by ensuring studies are planned, conducted, and documented in a uniform way. This produces high-quality data that regulators can confidently evaluate. Another major advantage is the mutual acceptance of data. When studies comply with internationally recognized GLP principles, regulatory authorities in multiple countries can often rely on the same results. That means fewer duplicate studies, faster product development, and less unnecessary testing, including reduced use of laboratory animals whenever scientifically appropriate. To achieve these high global standards consistently, GLP relies on 5 operational pillars. The entire GLP quality framework is supported by 5 core pillars: resources, characterization, rules, results, and QA audits. If even one pillar is cracked, whether it's an uncalibrated instrument or an unverified test compound, the integrity of the entire study comes crashing down during an audit. Over the next few slides, we'll examine how each pillar functions in our daily routines. Let's take a close look at our first foundation. Pillar 1, resources. Pillar 1 centers on resources, requiring secure facilities, qualified staff, and calibrated laboratory hardware. Your results are compromised. This pillar mandates that laboratory personnel are adequately trained, facilities are designed to avoid cross-contamination, and hardware is routinely qualified and maintained. Once our physical resources are locked down, we must focus on what we are testing. Pillar 2, characterization. The second pillar, characterization, demands full traceability and identity verification for all test items and biological systems. Do you know the exact purity, stability, and batch origin of the compounds sitting on your lab bench right now? What about the health status and lineage of the biological systems being used? Characterization ensures we know exactly what we are testing and what we are testing it on. If your test material is degraded or mislabeled, your entire dataset turns into a very expensive guessing game. With calibrated hardware and characterized test items ready, we need clear instructions on how to execute, which leads us to Pillar 3. Pillar 3, Rules, relies on pre-approved protocols and strictly enforced standard operating procedures. In a GLP laboratory, unscripted spontaneity is your enemy. Every task must follow approved protocols and standardized SOPs. Think of an SOP as a master recipe for reproducibility. It ensures that whether scientist A performs the assay on Monday or scientist B performs it on Friday, the execution is identical. Strict rules don't stifle scientific creativity. They guarantee that your data is repeatable and legally defensible. Executing these rules generates raw data, bringing us directly to Pillar 4, Results. Pillar 4 focuses on results, emphasizing legible, contemporaneous data capture and secure long-term archives. Data integrity lives or dies in Pillar 4. This pillar mandates that raw data must be recorded immediately, as it happens. No jotting numbers down on scrap paper to transcribe later. Furthermore, all raw data and study records must be stored in secure, fireproof, tamper-evident archives. If an auditor asks to see the raw balance printouts from a study conducted 3 years ago, can you retrieve them in 5 minutes? Finally, how do we verify that these first 4 pillars are operating correctly? That brings us to Pillar 5. Pillar 5 establishes quality assurance audits to deliver fully independent monitoring of all GLP activities. QA auditors operate completely independent of study execution, so they can evaluate procedures, facility logs, and reports without bias. They act as our internal safety net to identify and correct compliance gaps long before a regulatory inspector sets foot in our building. Now that we have covered all 5 pillars, let's review how this course is structured to help you master them practically. We've organized this learning program into 5 targeted operational modules. You won't just sit back and watch slides. You'll participate in interactive simulations, conduct mock deviation audits, and complete targeted comprehension checks along the way. By the time you earn your official certification, you'll have the hands-on confidence needed to defend data integrity in your everyday lab operations. Get ready to jump into our very first practical simulation exercise in Module 1.